Hello friends, how are you? I'm Solomon. I'm today with new video on chapter 3. And this chapter 3 contains about cellular base of life, and it is classified or divided into two parts. So in part 1 I'm going to discuss cell theories and cell organelles. Well, this is unit 3 of general biology course, and it focuses on the cellular base of life. In 16th, Robert Hooke, an English scientist, was the first person to observe cork with his crude microscope. His microscope magnification power was around 13 times, so he couldn't find the detail of the cell, but the cell cork. And there is no indication of the nucleus as well as other organelles. The first man who saw living cell under microscope, which had some improvement than that of Robert Hooke, was Antonio van Leeuwenhoek. His microscope magnification power was around 300 times. Therefore, he tried to observe living cells. For example, he observed in 1674 algal sporangea, which were living, moving unicellular organisms in a drop of water, and he called this organism animalcules, meaning little animals. Later, Matthias Sechliden in 1838 examined that especially all parts of plants are composed of cells. Then Georges Zechion in 1839 he examined that all parts of animals are composed of cells. Finally, German physiologist Robert Virchow examined this idea by containing that cells are arising from only pre-existing cells. By the way, the ideas of these three scientists used to put the cell theory. We'll see in the next slide what cell theory says. Okay, 3.1 is about cell theory. The following are some cell theories proposed by different scientists. For example, cells are, all organisms are composed of one or more cells. Unicellular organisms are made up of one cell, whereas multicellular organisms are composed of or made up of cells or many cells. The second cell theory is a cell is the basic structural and the functional unit of living organisms. A cell is structural unit because each organism is built from cells as a building built from different bricks. The organism's body is formed or built from cells. Therefore, structurally, organisms are built from cells. Functional units, because almost every chemical reaction that takes place in living organisms are occurred in cells. Therefore, cells are structurally unit for organisms as well as they are functional units of living organisms. The third cell theory is cells are arise from pre-existing cell. Each and every cell is arised from previously found cells. Therefore, all organisms are composed of one or more cells. A cell is basic structure and functional units of organisms, and cells are arise from pre-existing cells are some of cell theories that are proposed by different scientists. The other point is types of cells. Based on the presence or absence of nucleus and membrane-bounded organelles, organisms or cells can be grouped into two, based on the presence or absence of nucleus, as well as presence or absence of membrane-bounded organelles. Cells can be categorized into main groups. The first category is cells are eukaryotic cells. Eukaryotic cells are cells that have true nucleus, as well as they have membrane-bounded organelles. Prokaryotic cells are cells that has no true nucleus. They have no true nucleus, as well as they have no membrane-bounded organelles. So these two types or groups of cells are where cells are categorized. Bacteria and the blue-green algae are prokaryotic cells, where cells from other organisms like plants, animals, and like are prokaryotic cells. Eukaryotic cells. This is cell organelles. Cell organelles are organelles or parts of cell which have different functions. Even if the cell is small in its size, but it has different parts, or it has different organs which perform a specific function. So here, this is animal cells, and this one is plant cell. They have many organelles in common, but still they have some organelles are different, are found in plants, but that are not found in animals. To show here, cell wall, for example, is found in plant cells, but cell wall is not found in animal cells. For example, plant cells have central vacuole, whereas animal cells have no such parts. And also, plant cells have chloroplast, an organelle which helps to which used for photosynthesis, but animal cells have no chloroplast. Whereas animal cells have lysosomes, but plant cells have no lysosomes. However, plant and animal cells have many organelles in common. For example, both have mitochondria, powerhouse of the cell, and also both plant and animal cells have endoplasmic reticulum, either a smooth or rough endoplasmic reticulum. They have this organelle in common. And the plant and animal cells have nucleus, which controls the whole cellular activities, and they have microtubule in common, Golgi body or Golgi apparatus in common. Therefore, plant and animal cells have many organelles in common. In the next slide, I will try to discuss the functions of different organelles of cells. Typically, eukaryotic cells have three major parts. First part is plasma membrane, which is the outer boundary of the cell. It's found in the outer part of the cell, but some cells, for example, cells from plants, have additional outermost structure, which is known as cell wall. But this cell wall is non-living part of cells, as well as it's not found in all prokaryotic cells, meaning it is not a common structure for all eukaryotic cells. But almost all eukaryotic cells and prokaryotic cells, including prokaryotic cells, have cell membrane or plasma membrane. This plasma membrane is the outermost part of all cells that bound the cell from the outer or the external environment. The other part is cytoplasm. This cytoplasm is the structure or intracellular fluid which is found between the cell, plasma membrane and the cell, the nucleus. It is found between the nucleus and the plasma membrane. This is plasma membrane. This is cytoplasm. All the nucleus of eukaryotic cells are suspended here in the cytoplasm to perform their specific functions. Therefore, structure between the nucleus and the plasma membrane is the cytoplasm. The other is nucleus. Nucleus controls all activities of the cell, and most it is found or rests in at the center of the cell. The nucleus has different parts, and I will see in the next slide or in the next part these parts of the nucleus. However, plasma membrane, cytoplasm, and the nucleus are the three major parts of all eukaryotic cells. To define the term organelle, so an organelle is a specialized subunit within the cytoplasm of a cell that has specific function. It is part of a cell which perform different or specific function found in the cytoplasm of the cell. Generally, there are two types of cell organelles are there. First one is membranous organelle. These membranous organelles are organelles that bounded by membranes, whereas the other is non-membranous organelles. Non-membranous organelles are organelles that have no membranes that surround their parts. For example, endoplasmic reticulum, both rough and smooth endoplasmic reticulum, Golgi bodies, mitochondria, chloroplast, nucleus, lysosome, peroxisomes, and vacuoles are membranous organelles. Whereas ribosomes, centrosomes, cilia, flagella, microtubules, basal bodies, microfilaments, these are non-membranous organelles. Here, next is to discuss some organelles and their functions. For example, the first one is a nucleus. A nucleus is oval or spherical in shape and the largest central structure that's surrounded by double-layered membrane. For example, if this is a cell membrane that surrounds the cell, and this is a nucleus which is found almost at the center of the cell. So this nucleus is surrounded by double-layered membrane. This membrane that surrounds the nucleus is known as nuclear envelope. Nuclear envelope, it is double layer. Therefore, this nucleus contains DNA, which is the genetic material of almost all organisms, except some RNA virus. The function of DNA is to produce proteins, meaning protein is produced by DNA, which is found in the nucleus. Therefore, through these proteins that are produced by the control action of DNA, so nucleus control all activities of cells. So it governs or it masters cellular activities. As already said, the nucleus is surrounded by nuclear envelope, and it contains chromatins as well as contains nucleus. to discuss these parts separately. Nuclear envelope, which surrounds the nuclear material. If this is the nucleus, and in the nucleus there are different contents, therefore this cellular content, nuclear contents or nuclear materials are surrounded by nuclear envelope. So this nuclear envelope is double layer. It is a double layer membrane. It has two layers. Plus, this nuclear envelope has small holes intervally. These holes are important for exchange of materials between the nucleus and the cytoplasm. These perforated holes are important for the exchange of ions and water and other materials between the nucleus and the cytoplasm of the cell. The other part is chromatin. Chromatin means color materials. Thus, chromatins are chromosomes, and chromosomes are composed or made up of DNA and histone protein. Therefore, this DNA is genetic material which controls or which determine what kind of proteins are produced. Through this DNA, nucleus control almost all activities of the cell. The third part of the nucleus is nucleus. Plural form of this term is nuclei. These nucleus are engaged actively in synthesis of ribosomes. Ribosomes are organelles where protein is produced. However, they are formed or produced by the active engagement of these nuclei, which are parts of the nucleus. Cytoplasm is the second organelle, which is interior part of the cell, which excluding the nucleus. Meaning, if this is the cell membrane, and this is the nucleus, which is surrounded by nuclear membrane, the internal part of the cell, excluding the nucleus, is cytoplasm. This is cytoplasm. Interior part of the cell, excluding the nucleus, is a cytoplasm, and it contains different organelles that are highly organized and membranous organelles, which are suspended throughout the cytoplasm. Because they are specifically compartmentized, because each of these organelles perform different chemical reactions, or they carried out different chemical reactions. That's why it is said that cell is a functional unit of organisms. Different chemicals are produced in the organelles of the cell. Therefore, this cytoplasm contains these different organelles that are separately compartmentized to perform their own function. And these organelles in the cytoplasm are dispersed within the complex jelly-like marrow known as cytosol. This cytoplasm is filled with gel-like marrow, and it is known as cytosol. This gel-like structure, which is found or fill the cytoplasm, and it is known as the cytosol. Each organelle is separately organized, compartmentized, containing different chemical settings for fulfilling the cellular function. These organelles occupy about half of the total cell volume of the cytoplasm, and the remaining part is the cytosol. Half of the cytoplasm of the cell is occupied by these organelles, and the rest half is still cytosol or the gel-like marrow. The second organelle is endoplasmic reticulum. It is a fluid-filled membrane system present throughout cytosol. Here on this figure, you can see the endoplasmic reticulum. This is rough endoplasmic reticulum. By the way, there are two types of endoplasmic reticulums: rough endoplasmic reticulum and smooth endoplasmic reticulum. Their difference is at the presence of ribosomes on the surface of rough endoplasmic reticulum. Here, on the rough endoplasmic reticulum, there are ribosomes on the surface. However, smooth endoplasmic reticulum have no any ribosomes on their surface. This is the main difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. These rough endoplasmic reticulums are interconnected to the nuclear pore, nuclear envelope, as well as they are interconnected to each other. Each and every endoplasmic reticulum in the cytosol of the cell are interconnected to each other. This rough endoplasmic reticulum projects outwards the cytosol as stacks of flattened sac that are interconnected to each other. Here you can see each and every rough endoplasmic reticulum is interconnected on another. Rough endoplasmic reticulum is associated with ribosomes on its surface. There are number of ribosomes are found, so they are participated in protein production. Once protein is produced on the ribosomes, they are stored in rough endoplasmic reticulum. Some proteins are exported as secretory products. For example, hormones and enzymes are such kind of proteins. They are secreted and transported or exported to their target area. Some other proteins are transported to sites within the cell itself to use as a construction of new plasma membrane and new organelle membranes, because these cell membranes and organelle membranes have proteins as a constituent, because cell membrane is made up of lipid types, especially phospholipid. However, proteins are also found in such membranes. For example, lipoprotein. When protein is associated with protein, the lipid is associated with protein. Glycoprotein is when carbohydrates are associated with protein, as well as these transport proteins are protein types which are found in the cell membrane. Therefore, this type of proteins are transported to the cell itself, where such kind of membranes are constructed. Smooth endoplasmic reticulum is a network of interconnected tubes, because each endoplasmic reticulum is a tube that is interconnected to each other. Since this endoplasmic reticulum has no ribosomes, therefore they are smooth and they are not participate in protein production, but they have a number of functions. For example, they serve as packaging and discharging of proteins and fat molecules. Especially, they are found in most body cells. So they package and discharge this protein and fat molecules. All new proteins and fats passed from rough endoplasmic reticulum are gathered in smooth endoplasmic reticulum. Then portions of the smooth endoplasmic reticulum is bud off or pinch off and giving rise to transport vehicle, meaning whenever the protein and lipid molecules are reached in smooth endoplasmic reticulum, then they are packed by part of the smooth endoplasmic reticulum itself. Then after, this is used as a transport vehicle, and they are transported into the Golgi complex. Here, rough endoplasmic reticulum is here. It has ribosomes on its surface, therefore it is used. These ribosomes are used to produce protein. Once protein is produced on the rough endoplasmic reticulum, then they are transported into smooth endoplasmic reticulum. Then here in the smooth endoplasmic reticulum, these molecules are covered by the parts of or portions of the smooth endoplasmic reticulum and used as a transport vehicle. So they are transported into Golgi apparatus. Here in Golgi apparatus, further process will be carried out for further transportation. So this is another or one function of smooth endoplasmic reticulum. Next, this smooth endoplasmic reticulum serve as lipid metabolism because they contain enzymes for lipid synthesis, specifically cells that synthesize steroid hormones like androgen, testosterone, the like. Therefore, they serve as lipid metabolism. Next, this smooth endoplasmic reticulum serves in the detoxification of harmful substance. These harmful substances are either they are ingested into the body, or they may be produced as a result of metabolism. Specifically, such kind of smooth endoplasmic reticulums are found in the liver and the kidney cells. So they help to detoxify harmful substances in our body. And they serve in the process of muscle contraction. Specifically, such smooth endoplasmic reticulum are found in the skeletal muscle cells. Therefore, this smooth endoplasmic reticulum serve as a packaging and the discharging of protein and the lipid molecules. They serve in the lipid metabolism because they contain enzymes for lipid synthesis, and they serve as detoxification of harmful endogenous or exogenous substance, and they serve in the process of muscle contraction. These are main functions of smooth endoplasmic reticulums. The next cell organelle is Golgi bodies or Golgi complex, and this Golgi complex is associated with endoplasmic reticulum and contains a number of or sets of membrane bounded sacs called cisternae. There are sacs which are membrane bound. So they are specifically the number of cisternae depends on the cells specialized function. For example, cells which are specialized in protein secretion or protein production has hundreds of cisternae, but some cells only have one cisternae. As discussed in the previous slides, these newly formed protein and the lipids are part of smooth endoplasmic reticulum and directed and entered into Golgi complex or Golgi bodies, specifically to the cisternae. In general, these Golgi bodies or Golgi complex has the following function. For example, it is used in the processing of raw materials into finished products. For example, raw protein is finished into glycoprotein. As you know, this glycoprotein is made up of carbohydrates plus protein molecules. Therefore, this process is carried out in the Golgi bodies. And once these raw materials are changed into finished products, they are sorted and directed into the final destination. Therefore, the raw materials, the process of the raw materials are changed into finished products, and as the finished products are sorted and directed into their final destination are carried out by Golgi bodies or Golgi complex. These are the main functions of this organic. Well, lysosomes are another cellular organelles. Lysosomes serve as intracellular digestive system because they have a powerful hydrolytic enzyme. These enzymes are occupied in the sac, which is closed by membrane. Therefore, any unwanted cellular debris as well as any foreign materials like bacteria are taken into this organelle and immediately they will be digested and removed out of the cell. But the cell itself is protected or prevented from these enzymes by membrane surrounding it. Already these lysosomes are surrounded by membrane or they are enclosed by membrane. Therefore, enzymes in these organelles are not escape out of the organelle and affect the cell itself. Materials are taken into these organelles to be digested by lysosomal enzymes by a process known as endocytosis. Endocytosis can be phagocytosis if the material is taken in the liquid form, whereas the phagocytosis form is when large and multicellular particles in the solid form are taken into these lysosomes. The process is known as phagocytosis. Not only foreign materials like bacteria, but also old organelles of a cell can be taken into the lysosomes, and they will be broken down into their components. Therefore, any needed part of the organelle will be reabsorbed back to the cytosol of the cell, whereas the rest part which is unwanted part of organelles will be dumped out of the cell. The process by which worn out or old organelles are digested is known as autophagy. This is a process in which worn out or old organelles are digested by the lysosomes. In a condition known as lysosomal storage disease or TSH disease, lysosomes are not effective because they lack specific enzymes that are needed to digest any specific molecules. Therefore, this As a result, harmful waste products are accumulated and disturbing the normal functioning of the cell, and may result to fatal, or it may result to death. This disease is inheritable, which is related with genetics, or it is gene related. That means it is inherited from parent to offsprings. The next cell organelle is peroxisome. This peroxisome is a membrane enclosed sac, which is membrane enclosed sac, contains oxidative enzymes and catalase. This sac-like membrane enclosed organelle contains these enzymes, oxidative enzymes, as well as catalase that detoxify various waste products. For example, oxidative enzymes need oxygen to remove hydrogen from toxic molecules, for example, ethanol, which is consumed with alcoholic drinks, especially in liver and kidney, and then these molecules are detoxified. Catalase is an enzyme that an oxidative enzyme decomposing hydrogen peroxide into harmless water and oxygen, because this hydrogen peroxide, which is powerful oxidant, is toxic chemical in cells. Mitochondria is another cellular organelle, and they are important in energy production, so they are powerhouse of the cell. Energy in the nutrients is transformed into usable form of energy known as ATP. Later, we'll see how energy in the nutrients is converted into usable form of energy, which is ATP form, when we will discuss about respiration. However, this energy production is carried out specifically in the mitochondria of the cell. They are rod or oval in shape, and their size is almost equal to the size of bacteria. The number of mitochondria varies from cell to cell. Some cells may have hundreds or thousands of mitochondria, but some still may have a small amount of mitochondria. This depends on the requirement of energy for such particular cells. Mitochondria has two membranes. It is double membrane organelle. The outer membrane as well as the inner membrane are found in mitochondria. The outer membrane is surrounding the mitochondria, or the rest part of the mitochondria, and the inner membrane is used to form these foldings or shelves known as Cristia shelf-like or foldings which are found in the mitochondria. These cristia are filled with gel-like matrix, and this matrix contains mixture of hundreds of dissolved enzymes. Specifically, they are citric acid cycle enzymes, because these enzymes are used in energy production at citric acid cycle of respiration. Therefore, this cristia is filled with matrix, and the matrix contains these mixtures of hundreds of enzymes for ATP production. Second, mitochondria are unique organelles because of two things. The first one is this mitochondria are or have their own unique DNA, which is known as mitochondrial DNA. Second, this mitochondria can replicate themselves even if the cell which they are belong is not undergoes cell division. Because of these two things, mitochondria are unique organelles than any other organelles. First, they have unique DNA or their own unique DNA. Second, they can replicate themselves, even if the cells they are belongs are not in cell division. Well, chloroplasts are organelles which are found in plant cells, and they are highly participate in process known as photosynthesis. During photosynthesis process, water is split into hydrogen and oxygen. Then this hydrogen is joined with carbon dioxide and make carbohydrates. But oxygen is liberated or released to the atmosphere. So these processes are occurred in chloroplasts. They are found nearly to the surface of the cells because to receive enough amount of sunlight. If this is a nucleus, and chloroplasts are found nearly to the surface of the cell. This is to receive adequate amount of sunlight or for the process of photosynthesis. This is light. Therefore, they are found nearly to the surface of the cell. Plus, they are green in color. They are green in color because they have a green pigment known as chlorophyll. Therefore, they are green in color. By the way, we'll discuss in detail about parts of these chloroplasts when we'll see the process of photosynthesis in the next chapters. Therefore, for the time being, they are the site where photosynthesis are occurred, and they are found nearly to the surface of the cell, and they are green in color because of the presence of chlorophyll. This is enough for the time being. Vacuoles are another organelles which are found in plant cells, and they are formed by joining together of these many vesicles. When many vesicles are joined together, they make vacuoles. Their function is to keep or to maintain the turgid pressure of plant cells, because if this is a cell, these vacuoles contains different compounds or different nutrients or minerals. Therefore, whenever they get enough amount of water, they push the cell, the content will push or press toward the cell wall. Therefore, the plant will be, the cell will be turgid. Therefore, this keeps or maintain the plant to be rigid, as well as, for example, the plant's leaves will be flattened. But whenever there is lack of water, the cells become shrink. Looks like here. They lose the cell wall, which is the rigid part of the cell or plant cell, and they lose a cell wall, and the cells are shrink. Therefore, these vacuoles maintain the turgidity of plant cells. Whenever the plant cell obtain enough amount of water, the water will be absorbed and stored in the vacuoles, and after the plant cells will be turgid. Press all the cell contents are pressed toward the cell wall. So the cell wall is tough; it cannot be broken down. Therefore, it keeps the cell to be turgid. But whenever lack of water, the whole contents are shrink, losing from the cell membrane, cell wall, and the plants will be looks like dry. This is the main function of vacuoles. So this cytoskeleton is another organelle of cells. These cytoskeletons are complex protein networks. Actually, the word cytoskeleton is formed from two words, cyto and skeleton, meaning the skeleton of the cell. Actually, cells have no skeleton, real skeleton, but these proteins are working as skeleton. In organisms which have backbone or skeletal system, their shape and their size are determined by their skeleton. Similarly, the shape of cells and the size of the cells are determined by these protein networks, which are cytoskeletons. Similarly, organisms with backbone or with skeletal system, their movement is also determined by their skeleton. And similarly, these cells with cytoskeleton are affected, their movement is affected or determined by this complex protein network. Therefore, even if the cells have no real bone in the muscle, but their cytoskeleton, which is a complex network, a complex protein network, function as the bone and the muscle of organisms, meaning it provides the shape for the cells, it provides the size for the cells, and even it determines the movement of the cells, and all these functions are obtained or determined by the cytoskeleton of the cell. The cell. Anyways, this cytoskeleton is 4 distinct elements. This network has at least 4 distinct elements. For example, microtubule is one group, microfilaments, intermediate filaments, and the microtubule lattice are 4 distinct elements or distinct types of cytoskeleton. Microtubule is the largest, and the microfilaments filaments are the smallest of the group. In general, these cytoskeletons determine or provide, for example, the shape of the cell. The shape of the cell is determined by cytoskeleton. Some cells are spherical in their shape. Some are rounded in their shape, or some others have another shape type. Therefore, the shape of cells are determined by these protein networks. And also, this cytoskeleton provides structural support. So the structure of the cell is also determined by this cytoskeleton. Contents in the cells are organized by the cytoskeleton. Therefore, provides the organization of the contents of the cell. Substance, it provides the movement of substance through the cell, because molecules in the cells or substance in the cells can move here and there. Therefore, their movement is determined by these protein networks, as well as not only the movement of molecules in the cells, but also the whole cell itself. The movement of the whole cell itself is determined by this protein network. Therefore, cytoskeleton has a number of functions for the cells.